Hi, welcome to the Family Wellbeing Project. Thank you for agreeing to participate in this extra study that's attached to our transition study. Today we're going to talk about how to collect the DNA and RNA saliva samples. So before we start, when you receive the kit, you're going to receive instructions on how to collect the saliva, and you're going to receive two separate kits, one for you and one for your child. So the package indicated with the green stickers are for the mothers. The package with the yellow stickers are for the children. So each set contains the tubes to collect your DNA. The tube looks like this, and the RNA, the tube looks like this. So there's one for you and your child. Please remember to keep them color coordinated when you're putting the samples back in these bags. Again, the green is for the mothers, the yellow is for the children. When you're going to be taking your saliva samples, there are a few things to keep in mind. First, it's important that you do not drink, eat, smoke, or chew gum for 30 minutes prior to your saliva collection. When you're taking the sample, which we'll show you shortly how to do, it's really important to fill it to the line, okay? So when we say to the line, it has to be pure liquid and not just saliva bubbles. So do make sure that when you're collecting, you have enough that the liquid goes up to this line on this tube and to this line here on this tube. Okay. And just so you know, for the differences in the collection, which again you will see, to not get the tubes confused, for the DNA, when you're taking your sample, you're going to be spitting into this funnel. Once you have spit to the line with liquid, you will be taking the funnel top off very carefully and then placing the cap, which is also found in the box, onto the tube before shaking it. Today we're going to demonstrate how to collect your saliva. So both you and your child will receive a red box to collect RNA and a blue container to collect DNA. All of the information on how to do it and the tubes are in these boxes. Further, you will receive a document that explains all the steps in collecting the saliva as well. It is important to note that for 30 minutes prior to your collection, you cannot smoke, eat, drink, or chew gum. Further, it takes about 2 to 5 minutes to actually do the collection itself. It can be done, however, any time of day.
To help produce saliva, you can rub your cheeks in a circular motion. When you are ready to take your sample, open the tube. Be careful not to touch the plastic film covering the cap. Holding the tube upright, spit into the tube until the amount of liquid saliva, not bubbles, reaches the level shown. When the tube is full, take the cap and carefully place it on the vial and tighten firmly. At this point, the stabilizing liquid will be released from the cap. Then shake the tube vigorously for 10 seconds to mix the saliva and the liquid. Your RNA sample is now complete. Moving on to the next sample, open the box and hold the tube upright. You can again massage your cheeks to produce saliva. When you are ready, hold the tube upright and spit into the funnel until the amount of liquid again reaches the fill line, not the bubbles. Holding the tube upright with one hand, take your other hand and close the lid tightly over the funnel. This will release the liquid. Then, still holding the tube upright, unscrew the funnel top. Take the smaller cap that was found in the box and tightly cap the tube. Now, shake the tube for 5 seconds to mix the saliva and the liquid. Your second sample is complete.
So now that you've seen how it's done, you can see that it's pretty straightforward. So we'd just like to remind you of different things. So before you actually do the data collection, please remember to select the appropriate child in your household to collect the samples for the child. This should be the same child that you focused on during the visits and interviews that we did with you previously for the transition study. If you're unsure, please give us a call and we'll gladly help you out with this. Now to put it back, as you can see, I have my two kits for the parent and for the child. So what we'd like you to do to store it, so here I have the yellow, which is the child samples. You'll just put it back in the boxes, just like so. And put it back in the bag. Now remember, the DNA, okay, the blue tube had the funnel. You don't need to give us back the funnel. That can be thrown away. Just put the tube back, properly sealed in the bag. So these are the child samples, and you will do the same for your own samples. Just putting it back in and putting it back in the properly labeled bag, remembering that the green ones are for the mothers, okay? So once you do that, it'll be time to store it. These do not need to go into the fridge, unlike the ones you did for the transition studies. These can be kept at room temperature until they're either picked up or mailed to us. You will know which at the time of the study. So once again, thank you very much for your participation in our study. If at any point you have any questions, please do not hesitate to call us. Thank you.